You can now look at a Illumina video of the process. It's Illumina, produced by Illumina, and it will tell you about, you kind of get, there's other methodologies too for the generation, for the amplification of the fragments that you generate. But the Illumina, we'll go with the Illumina description of Illumina for the simple reason that we have an Illumina, and right now they are literally have set the standard in terms of the state of the art for this methodology. Okay, so we'll watch this video, we'll come back, and then we'll talk a little bit about the video and take it from there.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through Brij Amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After Brij amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated, similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities of next-generation sequencing.
So after seeing the movie, let's summarize the steps in next generation sequencing. And again, as has been said before, that was the Illumina channel method. Okay, there's methods involving microbeads which are used to amplify these fragments, which eventually become reads. Start out with a reference DNA sequence. Now, this is important because there is another type of a more challenging next generation sequencing experiment and data processing called de novo sequencing, right? That is where you do not have a reference genome and where you actually have to make sure not only that the reads are stitched together correctly, are put together correctly, but the order in which these reads are put together correctly is also very important. So de novo sequencing requires a lot more of processing power and a lot more of computational effort. So you take your DNA of interest and break it into approximately even-sized fragments through enzymatic methods or sonication, which is basically blasting it with sound waves. Then to both ends of your DNA fragment attach via the enzyme ligase an adapter, approximately 50 base pairs sequences, using polymerase chain reaction to make multiple copies of these fragments. And then you denature these fragments into single strands. Okay, that is kind of, you take the double strand DNA and you convert it into a single stranded DNA. So continuing the steps, you wash these single strand fragments over bead or flow cells. The Illumina method involves flow cells that contain primers that are complementary to the adapters. Now remember, this is beginning to look a little bit like the Sanger sequencing, right? That means the primer is the one that kind of creates the chain, which is kind of the beginning of the creation of the complementary chain to the reference, to the fragments that you've created from your reference DNA sample. So hybridization occurs since the adapters are complementary to the primers, and polymerase and nucleotides are used to generate double-stranded DNA. And when the double strand is denatured, the non-tethered strand, which is not ligated to the beads or the flow cell, is washed away. So Brij amplification is therefore necessary. One adapter hybridizes with one primer. The adapter at the other end hybridizes with another primer, forming a bridge, and that is what the movie showed you, right? So let's look at Brij amplification. So you have the adapter, which is attached to the beads or the flow cell, whatever substrate that you have, and then you have an adapter. And what you do is you create these DNA fragments, and then so this is, it's called a dense lawn of primers. Now you have an adapter which is complementary to the other primer, and what you see here is this kind of folds over, and the moment it folds over, the process of DNA polymerization using DNA polymerase actually creates a bridge, and this is the idea of a Brij amplification. Now, as soon as you see this, one of this is free, one of these is attached, and then once you wash this, okay, you're going to get one more. So this one folds over and begins to create a complementary double strand with this primer, with this terminus, and then you see the one with the pink folds over and attaches to another one, and then you kind of, that opens out into a single to another strand, and this is how you basically amplify, and this is called Brij amplification, and I'm sure the movie showed this more and more. So you see these little clusters, these clusters and repeated amplification. But this is the notion of Brij amplification, and you can look at this PDF from which some of these pictures are taken, and the movie also showed you this in better detail. So more, once large clusters of a single strand of DNA with an adapter at one end is attached to the primer at the other end are formed, then you introduce these primers that are complementary with the free-floating adapter of the single DNA strand sequencing along with reverse terminating nucleotides to which are attached fluorescent dyes. Reverse termination is the same as the DDATP from the dideoxy ATP methodology from ATP or GTP or CTP or TTP from the Sanger sequencing, which allows the adding of only one nucleotide at a time. So this is the reverse termination step. And so this is the Sanger sequencing method, except done several 100,000 times over on the same substrate. At the addition of each nucleotide, a picture is taken, and the color of the picture tells you which nucleotide is added because they fluoresce differently. And so what now you also, in addition to just understanding what's going on, the dyes are differently colored, so they fluoresce at a different wavelength. They indicate what nucleotide is added. Now, what you have to understand is you already begin to see why this becomes a pretty challenging thing. Because remember, you're creating several hundreds of these, several hundred thousands of these, and at the addition of each nucleotide, where there is the complementarity, you begin to get a picture. So if you're going to see so many of these nucleotides being added, there's going to be pictures taken at every step. So each of these colors is an indication of what nucleotide is being added. And so for that exact same spot, you begin to see kind of the succession of pictures, and the succession of colors tells you that this is how the sequence development takes place. Now, remember, I told you at the beginning that processing is okay with high-performance computing, with supercomputers. This is possible, but data storage is a problem. So think of this experiment where at every step, hundreds of thousands of these in these channels or the microbeads, when each nucleotide is added, you take a picture. And then you take another picture, you take another picture. So all this data has to be stored. Think about essentially a movie, right? And this is a good analogy. So you have, let's say, you take really what is a movie? A movie is a collection of photographs that are kind of threaded together. Okay. Now, what is the difference in terms of the size of you take a picture on your phone, it's about a megabyte, right? Now you take several instances of these photographs of a particular event unfolding over 5 minutes, because remember, if you want pictures to be threaded into a movie, they have to go within the persistence of vision, right? 1/8 or 1/6, the picture remains 1/6 of a second. So if you want that to not appear like a movie and not disjointed, that's 1/6 of a second. So 6 pictures a second, that's 6 megabytes per minute. That's 360 megabytes, right? And so essentially for a 5-minute movie, you're going to go over a gigabyte. Now think about that kind of a picture that's taken for an entire experiment. You take it at the addition of every nucleotide, and for one particular experiment, now you can begin to see, and these pictures are not necessarily 1 megabyte big. These are a lot bigger. And how much effort do you need to put in terms of the data storage, and what kind of thinking goes into the data storage? And that's important. Okay, so what you see here is that you see a picture that's taken, that this nucleotide, so you know exactly at which spots in your lawn, for example, where your DNA sequences are being developed. So this fluorescence picks up some a color, another color, another color, another color, and you notice something, that obviously they're not the same, because at any point in time a nucleotide is added, is a different nucleotide, right? And then what you do is you create a composite of these pictures. Okay, now this sequence that's now created at a certain fragment where the reverse termination stops it, kind of like the dideoxynucleotide stops the Sanger sequence from a sequencing process from proceeding. you see the sequence, and this sequence is now called a read. So now once you have these reads, putting these reads together into a coherent sequence, remember now that's, so you have a read. This is where the Sanger sequencing stopped. At the Sanger sequencing, now you had little of these fragments and you kind of put them together, right? Now expand this exponentially. These reads are 20 to 50 nucleotides long. Now you've got to take each of these reads and stitch them together at a genomic level. Okay, so the sequencing process following next generation sequencing involving assembling the reads into a coherent sequence. Now that is why this is I started by saying next generation sequencing at this point in time is synonymous with bioinformatics, which means most of the bioinformatics efforts, and we also discussed how bioinformatics evolves. So most of the bioinformatics efforts actually goes into putting these reads together into a sequence, so you begin to get a sense of what the genome is like. Okay, so let's say, for example, how do you put these together? So I have a read, I have another read, I have another read, I have all these reads that come out of my next generation sequencing experiment, and this is a consensus sequence, also called as a reference sequence. So I begin to see here, for example, there is a TTG, and this starts with a TTG. So I said, well, this TTG could be the same, TGG could be the same, and yes, it's quite the same, right? TGG. Then I have another reader. Remember, these are randomly broken, using the enzyme, and so enzyme or solid sonication. And so now I see a TGG, so I can kind of line up my TGGs, and then there's some other parts. So you line these up during the different reads, and then you get a consensus sequence. Now here, this is consensus C. There is a possible error. There is something that needs to be taken care of. So these are not always perfect, right? And so this is the consensus sequence you get by putting these reads together. Now think about it as hundreds of thousands and 100,000 of these reads. They have to all be assembled, matched, assembled, matched, assembled, matched until you begin to see the patterns that kind of overlap. Okay, that's kind of the sequencing process. And you can see the same thing. We have a consensus sequence. This is the multiple copies of the genome. You by sonication or some sort of a, you fragmented these. Now each of these fragments went through a next generation sequencing, the experimental process, and you have all these reads now. These are all the fragments that are created. Now, for example, in a particular part of the genome, there is a low coverage. Okay, so you don't have as much, which means that when you try to put these together, in a low coverage situation, you realize that, well, you have a greater potential for error because you have fewer reads to match, right? And so what you see is you see a read, and this little part of the read of this particular read matches this particular part of the read. So you say, okay, I can put these together, I can put these together. Now think about putting this together computationally. And that is where the challenge occurs, right? And when you put this together, you get the consensus sequence, okay? Now, the data handling, right? So you saw those pictures, and I talked data handling can take place using high performance computing. If you're doing a de novo sequencing process, then of course it's a lot harder because you do not have a reference to match it against. What are the file structures? You can have text files, you put it in Excel spreadsheets, you can have databases, you have these picture files, these big gigantic picture files.